and welcome everybody who dialed in. I will not take time a lot from the presentations. Just as an announcement for the seminar series, I think we are experiencing a time where we do not do only alternatives to animal testing, but this is kind of radiating into many areas, and the techniques will be used in many biomedical fields as such because they are so valuable. And I think antibodies and the generation of antibodies is one of the major examples. So alternatives to animal is one driving force, but I think there will be hundreds of broad applications in many fields. And I think we will learn a lot from these two talks today. And I think with this, we start. And I think, how was it? Was Pierre starting or Alison?
Alison will start.
Alison starting, ladies first, that's right. Okay, Alison, go.
Okay, thank you. If I could just take this opportunity to introduce the non-profit organisation Affability, AFFA being the acronym for Animal Friendly Affinity Reagents. So Affability is striving to replace antibody production methods that are animal-derived with those that don't require their use, to not only reduce our reliance on animals, but also to encourage better science and positively impact society. Affability does this by using a multi-pronged approach, creating awareness around all aspects of antibody production by challenging the enforcement of Directive 2010/63 EU on the use of animals in scientific procedures, and by improving accessibility to replacement methods and resources. Today, I would like to discuss the changing landscape for animal-derived antibodies, or ADAs. ADA production is an animal welfare and scientific concern that's flown under the radar for decades, even though it's an obvious candidate for review in accordance with the EU Directive 2010/63 EU. Now we're seeing a big sea change, one that affects all scientific disciplines considering how much antibodies are relied upon. So understandably, the changing landscape leaves many questions and concerns. So as an introduction to this session, I will briefly discuss the antibody issues that have motivated the EUR-LECVAN recommendation released in May 2020, address why we were not already using replacement methods, the challenges now facing the scientific community, and how our approach as end users, developers, suppliers, or project license authorizers should now differ. So, to make sure we're all on the same page, when I'm talking about animal-derived antibody production, or ADAs, I'm referring to all different antibody production methods relying on animal immunization. So the natural mammalian immune response is triggered when a foreign invader, known as an antigen, enters the body, to which the immune system launches an attack to neutralize it. Now, scientists observed this more than 100 years ago and found that if they injected an antigen of interest into an animal, it would be recognized as foreign and an immune response would be launched against it. As part of the immune response, antibodies are naturally generated, and these could be extracted for use in future tests. For example, to develop a diagnostic test, a drug therapy, tests for the illegal use of performance-enhancing drugs, fertility tests, or to detect food or environmental contaminants. So the animal is immunized repeatedly to obtain higher titers of antibody specific for the antigen, and that's in combination with adjuvant, which boosts the immune response, and then blood samples are taken to monitor its progression. Smaller animals are sacrificed to extract an adequate volume of blood or to dissect the spleen. Larger animals tend to be used repeatedly for continuous production. The animal's response can vary from mild to severe and localized to systemic, but since the antigen may be novel, the animal's response to the immunization will not be known in advance. Now, the antibody production industry uses around 1 million animals per year, and that's mice, rats, rabbits, or larger animals, which are mostly ungulates, and these are animals with hooves, such as goats or horses. So they can be produced as polyclonals, where a cocktail of different antibodies all recognizing different sites on the target are collected from the blood, or they can be produced as monoclonals, where they are collected from the spleen, and here you can collect an antibody against one specific site rather than a cocktail of different ones. Both these methods lend themselves to conversion to recombinant format in order to alter characteristics of interest, since the antibody DNA from the B cells from the spleen or circulating in the blood can be cloned into bacteria. This is also the format for animal-friendly antibody production, cloned from synthetic genes or non-immunized human donor B cells. Hence, if you hear or read the word recombinants, sometimes even linked to the word animal-free, don't assume this automatically means animal-friendly. It may actually still be animal-derived. Enzo Life Science Group, for example, advertises recombinant antibodies as animal-free, but their criteria for classifying their antibodies in this way is different because the antibodies are actually mostly all derived from animal immunization. Unfortunately, we've been relying on animal-derived antibodies for many decades, aware and resigned to the fact that they have many disadvantages that hinder research. Antibodies are seen as a reagent or a tool to answer a pressing research hypothesis. Hence, people want to buy them, they expect their quality to be good, what it says on the tin, so to speak, trust the answer that they give, and don't expect to spend the duration of their funding period or PhD problem solving. However, there are hundreds of articles complaining about their quality, and sometimes it's years of research and citations before the fault is identified. This has a resounding impact in terms of wasted cost, time and resources, a reported annual loss of $800 million to the biomedical research community. There are also repercussions on diagnosis and health management, which is simply not something that the research community, publishers or funders should be tolerating. Therapeutic antibodies, of course, are required to be fully characterized in order to meet regulatory standards and so do not share these flaws. I mean, to paint a realistic picture, there are some positives. Polyclonals are inexpensive and quick to produce in a matter of months. An endearing characteristic is that they recognize multiple sites on the same targets, so you can imagine how this might be beneficial, for example in developing a drug against a highly mutating virus where the original site recognized by a single antibody might be lost over time. However, in the majority of applications, this can be a disadvantage since there is a high potential for cross-reactivity and misleading data. There's also the disadvantage of batch-to-batch variability, since no two animals ever produce an identical immune response or even the same animal to the same antigen injected more than once, which leads to irreproducible data. And there's no easy way to access or store the genetic information to ensure reproducibility. Monoclonals, on the other hand, are more specific, recognizing only one site within the target molecule, so there is presumed reduced probability of cross-reactivity. However, we now know that there are issues with genetic drift and contaminations by cells containing other genetic sequences, and that they are therefore not as specific as we originally thought. Immunized recombinant antibodies do share many of the same advantages as animal-friendly antibodies, or AFAs, since they are at this stage also genetically sequence-defined. But it is a significant time investment to get them to that point, and there is no net reduction in animal use. Also, some of the problematic issues are inherited from monoclonals and polyclonals, and as I said before, they are difficult to identify in catalogues. A few companies are now finally taking measures to improve the quality of their animal-derived antibody catalogue by transferring their stock antibody gene sequences to recombinant format. However, this approach is time-consuming and costly, therefore not likely to be done by the majority of antibody providers. So how do animal-friendly affinity reagents compare? I refer to these as AFAs, or they are otherwise known as non-animal-derived antibodies. So using a process which replicates the natural immune system, a library is developed in bacteria containing billions of antibodies that all recognize different targets. The source of this library is replicated non-immunized human B cells or synthetic antibody DNA sequences. You only have to make this library once, and it is then equivalent to a lifetime supply of animals. There's no immunization or waiting around for an immune response to develop. During the development of the library, right from the start and at every stage of the development process, the genetic sequence is routinely accessible for engineering. For example, this has a positive impact on antibody therapies. Since the gene sequence is known even prior to selection, undesirable characteristics such as those that cause developability issues can be filtered out at an early stage. The diversity of different antibodies in a well-designed library is also similar to that found in mammals, so the probability of one of them finding the right target during a process called panning is very high. Once the antibody against the target of interest has been selected, it can always easily be identified by its sequence, and this means unlimited reproducibility and the potential to further refine it. Since animals are not involved, it is possible to work on difficult targets such as toxic or pathogenic antigens, and even a cocktail of antibodies can be made to recognize different epitopes on the same target, which is an attractive quality of polyclonal antibodies. In fact, so good is this technology that it was recognized with a Nobel Prize in 2018. So this brings us to the Earl-Ekman recommendation released in May 2020 on non-animal derived antibodies. An Earl-Ekman recommendation provides the views of the JRC's EU reference laboratory for alternatives to animal testing on the scientific validity of alternative test methods, and this follows the legislation set out in Directive 2010/63 EU on animals used in scientific procedures. Now, all the information I've shared with you today can be found in this document. The Earl-Ekman Scientific Advisory Committee's endorsed opinion was that there is sufficient evidence to satisfy that non-animal derived antibodies are mature reagents generated by proven technology, and that due to their ease of manipulation, non-animal derived antibodies offer significant additional scientific and economic benefits. Then that in terms of addressing barriers to uptake, there is a need to promote accessibility to non-animal derived antibodies within the research, diagnostic and regulatory communities. Then the ESAC's unanimous conclusion was that based on the scientific evidence, non-animal derived antibodies are able to replace animal derived antibodies in the vast majority of applications, and that means all known applications. And that moreover, well-characterized recombinant affinity reagents will improve the reproducibility of science and positively impact society. Taking the ESAC opinion into consideration, the Earl-Ekman independent recommendation was that animals should no longer be used for the development and production of antibodies for research, regulatory, diagnostic and therapeutic applications whenever a replacement is possible. One opinion that I've heard is that the recommendation is premature because there are areas that still need attention. Whilst it is true that there are areas that require attention, it's not the case that the recommendation is premature. The replacement technology has been around for 25 years or more, and this was described in an Earl-Ekman workshop back in 1997 and is widely publicised, so if anything, it's overdue. The recommendation recognises that additional measures are needed, i.e. funding, education, accessibility and availability, and that member states need to implement these supporting factors. Because of these factors, the transition just wouldn't happen within an acceptable time frame without the motivation that the recommendation has provided. So the point of the recommendation is to motivate the transition that was not happening naturally. So with all these advantages, the obvious question is, why were we not already using replacement methods? The reason why this technology has been overlooked for decades is that it's largely believed that the technology is not yet mature enough and ready to replace animals, and that affinity is poor, or that a whole animal immune response is required to develop antibodies, or that these are not a good replacement for the polyclonal characteristics of multi-site recognition. And then the most contentious issue is with therapeutics, where it's believed that AFAs suffer greater developability issues, or longer lead optimization time, or run the risk of greater attrition rates and ultimately reduce clinical success. And you can find such comments in the non-technical project summaries available on the EU website. With the exception of therapeutics, the scientific misconceptions are addressed in the working group report that accompanies the EULEC-VAN recommendation. Now, it's obvious that there's been a clear lack of understanding of this new sophisticated technology, and it remains shrouded in mystery. A different skill set is required compared to animal-derived antibody methodologies. So there's been a lack of expertise and a lack of motivation by the users of traditional methods, and a propensity towards familiar methods due to the cost and time required for implementation. The replacement methods have also largely been used in the pharma sector, attracting high prices, royalties, and state-of-the-art high throughput processing, and this is immensely off-putting for academia or the commercial sector. Antibody therapies are quite a different ballgame, where cost increases exponentially and demands on their functional requirements are higher. So beyond antibody selection, regardless of their source, there are many choices made possible by different antibody engineering strategies. Here's a table illustrating the main phage display or AFA libraries known to the therapeutics industry. There are claims that the higher number of animal-derived antibodies used in therapeutics is testament to their superior quality, and for an updated list of antibody therapies that are in review or approved, the Antibody Society website is a rich information source. The truth is, we don't really know the reason for this, but it's a massive leap to reach such a conclusion when we don't know anything about the scientific approaches or financial commitments made in a particular drug development program prior to clinical outcome. The most recent analyses indicate that both platforms have a similar success or failure rate, so it's quite likely that what we're looking at is the result of a good or even aggressive business development strategy to gain a strong foothold in the market. Or, of course, we know that people prefer to go with technologies that they are most familiar with. Here are some examples of the patents that were in place but that have now expired. So bearing in mind that drug development from phase 2 clinical trials to approval can take 7 years, and from initial development to preclinical around the same length of time again, it's not surprising to see some delay in regulatory approvals. So all we can really say with any conviction is that AFAs are making a good impact and gaining regulatory approval in the therapeutic sector, and that at the moment they're in the hands of relatively few groups. So in light of the EULEC-VAN recommendation, the scientific community faces many challenges, some of which are very tricky. A project license application is likely to be refused, and I hope that now there will be increasing pressure to use sequence-defined, identifiable, animal-friendly antibody resources. But accessibility and abundance of resources is currently limited and more expensive, at least until demand increases and competition heats up. There is a cost and time commitment for implementation of resources, and is there financial or expert support for this? Also, the research community has to be motivated to source animal-friendly affinity reagents. In light of these issues, the problem could simply be driven away instead of being resolved if we don't take action, and that is a huge problem. Then, of course, there is concern about the socio-economic impact during the transition period and a reduction in the research space. So there are a number of solutions to facilitate a smoother transition. A lack of resources does not justify animal use. These are attainable, so they have to be implemented before identifying any case for animal use. For example, equipment, services and consumables. Phage display library development and selection from that library can be performed at a very reasonable cost by competent experts using standard molecular biology techniques and no specialist equipment. The time investment in library design is also worthwhile. However, there is of course the potential to invest in higher cost state-of-the-art facilities or purchase a license for a bespoke library, for example in the therapeutics arena. This costs as much as a breeding pair of licensed transgenic animals used to produce humanized antibodies. Resources also cover adequate training of personnel and access to facilities from an individual lab or from a centre of excellence or commercial resource. Of course, not every lab has to suddenly become an expert, and now maybe the resources will be available in different labs to the ones that were previously relied upon for antibody production, and that includes bioinformatics support. I believe that support from all levels is required here. Financial incentives, subsidies, grants, publishing and regulatory support. Otherwise, animal-derived antibodies will simply be outsourced. I think this is where much of our focus should now be. We want to see a commitment to the 3 Rs, but on a practical level, how can we do this if not supported by funding calls or other financial incentives? We need a boost in this area to improve accessibility, resources and training of personnel. So then the 3 Rs can be applied, having established that the above mentioned factors are in place, where it can be ascertained if in vitro antibody development failed for scientifically unresolvable reasons on a case-by-case basis. And if failure is the case, does this mean that animal-derived antibodies therefore are scientifically justified, as the reason for failure may not justify animal use? What do I mean by this? I refer you back to my earlier slide. Animal-derived antibodies may also fail, so they're not always a solution to failed in vitro projects. It's always important to point out that all antibodies are biological molecules, and so they're not perfect regardless of their source. Also, quite often it's the antigen that presents the biggest challenge, so actually this is like to be a problem-solving expedition. Recent work published in Nature regarding the use of a clever anchoring system and a functional approach with cell sorting to identify GPCR antibody agonists is a fascinating example of this. Other important considerations are: could other methods of analysis such as mass spec, PCR or reporter assays be used to provide the data required? And then, bearing in mind the flaws inherent in animal-derived antibody approaches, justification that there is added value in using animals at all in view of the information that can be gleaned from other types of analyses and the ultimate aim of the project. And let's talk about the wastage. Something else that's overlooked. Is there actually a need for yet another antibody? For example, there's more than 900 antibody clones for the p53 protein and around 8,500 for epidermal growth factor receptor. And with that, I will finish and leave you with some literature resources and say thank you very much for listening.
So thanks a lot, Alison. This was extremely informative and extremely well prepared. I guess you have done that many times, so it's good that the audience can profit from this. So I think we will wait with the questions then for both talks. And I will just kind of introduce briefly Pierre Cosson, who will tell us more about the practical production, I guess. He's one of the facilities doing this. So Pierre, please just continue.
Thank you, Marcel. Let me share my presentation. Is this okay?
Yes, good.
Good. So on this first slide, please note I'm putting the coordinates where you can reach us, and I encourage you to. The aim of this presentation is very pragmatic. I'm trying to convince you to do something now. A very short introduction on the fact that most published research findings are false. This is not a personal opinion. This is the title of a paper from 2015. And it's stating something which has become pretty obvious, that there is very low reproducibility in life science. Something like 10 to 50% of published results can be repeated. 10% repeated results means 90% non-repeated results. It is quite frightening. There are two main reasons for reproducibility problems. One is problems in the experimental design and the analysis of the results. And the other big problem is problematic reagents. Among the problematic reagents, the top is antibodies. Antibodies are used by a huge number of labs, but fact, about 50% of commercial antibodies do not recognize their intended target, or they lack specificity. And so whenever you order from a very nice catalog and you get your antibody, you have 50% chances not to get what you think you're getting. And there's no way to know in advance. And you've heard that recombinant antibodies, meaning antibodies that are monoclonal and sequenced, should replace traditional antibodies for a lot of good reasons. And the main part of this talk is dealing with what do we expect of antibodies and how can we get this? So to allow reproducible experiments, antibodies must be, number one, clearly identified. You have to be able to say, I'm using this antibody so that if I want to reproduce your experiment, I can use the same antibody. They must be available. You must have access to them, and I must have access to them if we are to make similar experiments. And they must be fully characterized so that we know how and when to use them. And let's start with the identification. And I took one example actually, and to be honest, initially I thought that would be my easy control. Antibodies against the MIC tag. It's one of the most used antibody in literature. It's one of the most used protein tag. Many people know what the MIC tag is. Okay, you type MIC tag in SciTab, which is one of these areas where you can see commercial antibodies. There are 1,800 commercial antibodies that recognize this short stretch of amino acids. 537 of them are polyclonal. As we've heard, they are highly variable from one animal to the other. They're present in limited amounts, they're present for a limited amount of time, and they belong in the trash. There is no way you can do reproducible experiments with polyclonal antibodies. I'm not saying polyclonal antibodies don't work. I'm just saying if you use a polyclonal antibody and I use a polyclonal antibody, we can expect not to get the same results. 1,300 of the antibodies available are monoclonal. And if you start looking, you get a huge list of names. Is 9B11 the same as 71D10? You don't know. It's just a number in a catalog. So you don't know if these antibodies are all different or all the same. If you take 9B11, for example, there are 14 antibodies sold by 3 companies that have this name. And by the way, if you look for 9B11, there are 8 other antibodies named 9B11 that recognize completely different antigens. 9B11 is not a good name, it's too short. It's been used by many different labs to name completely different antibodies. So it's creating a little bit of a mess. Now, if you're familiar with the field, 9E10 is the most well-known anti-MIC antibody. So 9E10 is sold by many companies. There are more than 500 antibodies available. So at least you would think if you use that one, we're all using the same one. But actually that has been tested. 3 groups have sequenced the 9E10 antibody. And actually, amazingly, the 3 groups came with 3 different sequences. They differ by 6 amino acids, which can be maybe not important or maybe super important. You don't know, I don't know, nobody knows. And by the way, there are other sequenced anti-MIC antibodies. And the best one is TA002. So summary, my anti-MIC is not your anti-MIC antibody. And my 9E10 is probably not your 9E10. You can imagine the problem for reproducibility. So to provide clear identification, antibodies must be monoclonal, they must be sequenced, and they must be classified. It's useless to know that somebody sequenced an antibody and has the sequence if you cannot get access to the sequence. How do we address this? We have created a database where we are gathering all the antibodies with published sequence, which are available in the literature or available from our discovery facility. Of course, that's much less in the literature. Today, there are 24,000 antibodies listed in this database. You want to know if your favorite antigen, there are recombinant antibodies against it, go to the database, check it. And by the way, and this is a call for help, if you have antibodies for which you know the sequence and are not in the database, contact us, deposit them. We can make it easy, we can make it worthwhile for you. It's really too bad to keep antibodies and not to share them with others. So if you type, for example, in the database, this is the first page of the database. If you type insulin, you find that there are 20 hits, so 20 entries in the database. Each of these entries has received a unique identifier, so AE804. It corresponds to one sequence with one single ID. So by the way, that means that if you and I use the AE804 anti-insulin antibody, then we know that we're using the same antibody. Reproducibility. The second problem with antibodies, recombinant antibodies, is availability. Okay, there is an AE804 antibody that recognizes insulin. How can you get access? How can I get access? And what we have created linked to this database is a production center. So we really worked at the procedures to be able to produce any of these 24,000 antibodies at a low cost. And I heard Alison mention that cost of recombinant antibodies is an issue. These are the cheapest antibodies on the market. And you can order any of these antibodies. These are the coordinates of the antibodies. If you go to the page, not only you have a search where you can search for your favorite antigen, antibody, whatever, but we have also identified some of the favorite antibodies that we have tested and that may be of general use. So for example, we have selected the best available antibody against the HA tag, the flag tag, the MIG tag, GFP, compartment cell markers, Golgi, mitochondria, endosomes, cytoskeleton, pancreas, insulin, glucagon, PPY, and then exotic fields like dictyostelium, discoideum. So all these are available and they can be produced, and they're usually produced as mini-bodies. A mini-body is just like an antibody, except that it's a bit simplified. It's only one chain. It works exactly like an antibody. You would not make a difference, except that you can change the constant part, which is here, so that an antibody can be produced as a mouse antibody, because it has a mouse FC, or as a rabbit antibody, because it has a rabbit FC, or human, or a guinea pig, or a rat antibody. So you can choose the format you want, which can be very useful if you want to do double immunofluorescence or triple immunofluorescence. Finally, antibodies to be useful must be characterized. And I mean fully characterized. And just a word, what is it to characterize an antibody? Number one, each antibody has to be characterized. It must be done for every application. So does this antibody work for immunofluorescence? Does it work for Western blot? It must be done with every protocol variation. Maybe it works in paraformyl fixed cells, but not in methanol fixed cells. This is really important. We need to know that. And all this knowledge is useful only if it is published, which means accessible to everybody. So this is why we actually created a journal, a scientific journal, which is dedicated to characterization of antibodies. It's an open access journal. It's dedicated to validation of recombinant antibodies, only recombinant antibodies. And it's really one antigen, one technique, one article. So this is a typical article. These antibodies with these reference numbers recognize GST by Western blot. Or here are two other papers. This one says that AF122 recognizes its target by immunofluorescence and describes the technique. This one compares 6 different antibodies against insulin to define that the best seems to be AE804. I'm about to conclude, and I'll conclude with another citation from Greta Thunberg, that one, blah, blah, blah. It's been now at least 10 years that people are arguing that recombinant antibodies should be used because they're scientifically desirable. But now the tools are available. Any of you can order any of these antibodies. But it's not going to work if everybody does not make a little something. It's a collective enterprise to produce these antibodies that we can do for you, to characterize them, to use them, to see when they work, when they don't work, to publish this. And I'm really inviting you to do this. Either we will do this as a collectivity, or we will keep relying on catalog numbers of companies, not knowing what is hiding behind the catalog numbers. So it may be a small individual effort to get into recombinant antibodies, because you will have to characterize the antibody the first time you're getting it. But in the end, this is good for you. You will get good science. This is good for the community. They can reproduce your experiments. And with that, before concluding, I'll just mention here, listed on the right, all the foundations that have supported this work. I hope I can answer your questions. Thank you.
So thanks a lot. This was very nicely complimentary and really a call to do things. I also get a bad conscience that we haven't used your antibodies. So how do we do, Georgia? You wanted to use the questionnaire at some point?
Yeah, so Silke can maybe start the poll. So we use this time also so you can think about your question and use the Q&A chat. And yeah, we thought to have indeed some really to fill the room and see how much you are aware of what you're using and of alternatives around. So take the time, please, to reply to the poll. Silke, let us know when you see enough replies. And I see that someone had raised the hands in the participants. We cannot allow vocal question, so I don't know if it was for mistake. Otherwise, please write it in the Q&A. So I'm not sure if the panelists, you can also see the replies while they're coming. Okay, so then maybe we can already have an idea. Perfect. See a question here.
I'm very encouraged that everybody knew that they were animal derived, because quite often people are not really considering this at all.
Yeah, we also thought, okay, indeed, it's a good question. Maybe we give it so for granted, but so we are almost complete. Maybe Silke, we can share the results. So we had, yeah, as you say, Alison, 100% knew that were animal derived. But it's also true that most didn't use non-animal derived antibodies, quite a number. And yeah, we also were curious if most of the participants were buying the antibody, or they are like Pierre and able to produce them. Okay, and we also, as you mentioned in your presentation about some of the issue about reproducibility, it's quite heterogeneous. Sorry, a bit. And yeah, and the last question, if they found if the specification were easily accessible, and here I would say that okay, most of people were quite happy with the information they can derive. I don't know if you have already some comments about what you saw here in the polls, Pierre or Alison.
On the issue of reproducibility of experiments, I think there are different issues really. It's one thing to have an antibody and that it works repeatedly, but if you're thinking reproducibility at a larger scale, your problem is to reproduce, let's say, the experiments done by somebody else 10 years ago, then you reach a different type of problem, which is, can you get the same reagent? And so there, it's not just did the antibody work and work reproducibly, but can you get the same antibody? And that's really something which we tend not to consider in our everyday experiments, but becomes a big issue when you're trying to reproduce existing work because you want to extend the result, or because you want to contradict it, or because you want to test it in a different way.
Yeah. So I see a question in the Q&A that asking, and maybe I can put it live, how easy it is to produce non-animal derived secondary antibody?
It's a very good question. It's not more difficult than to produce any antibody, and in principle, in the coming years, we should replace secondary antibodies with recombinant antibodies. But if you think of it carefully, it's quite a technical challenge, because you want to have antibodies that are, let's say, that recognize mouse antibodies and only mouse antibodies to be used as a secondary in a mouse when you're using a primary mouse antibody. You want to be sure they don't cross-react with other species, but then you don't know whether you should have a mix that recognizes all the different antibodies from mouse, or each different antibody, IgG1, IgG2A, IgG2B. You don't know if you want the same thing for every source of antibodies. So some companies are starting to work on that. And for example, Apcalis has described this first mixture of, so it's a mixture of antibodies that are specific for, let's say, mouse antibodies, as opposed to rabbit antibodies and other antibodies. But it's not fully available now. I haven't seen these reagents already coupled to fluorescence, coupled to secondary reagents. So it's coming, but it's not quite there yet. Would you agree, Alison?
Yes and no. I've been watching these closely because I like to use them myself for all the obvious reasons. And I have been in touch with Apcalis, and they've sent me one of these, so they recognize the FC portion of a primary antibody, just to specify that. And they did send me a sample of one of these multiclonals with a tag. So I think just communicate with them, and they'll be very accommodating to try and help you. Also, of course, I didn't actually see that question myself. It's not coming up in my chat, but I understand the question was about whether or not you can have animal-friendly secondary antibodies. But of course, you also have to consider that you might want to have a primary antibody with a tag that you could recognize with a secondary antibody, rather than the FC portion, which does make the situation easier, I should imagine. As long as it's not 90-10, maybe. Then you can try and find a secondary antibody to one of the tags. And also, you can use assays that are not antibody based to detect some of these tags. For example, if it's a his tag, you can use a NIC or NTA system, which is conjugated to recognize that. There's a strep2 tag alternative that doesn't involve antibody use, etc., etc. If anybody wants more information about those, then contact me and I'll give you more information about them.
Thank you.
I fully agree with that comment, and I should say, in general, Alison prefers to refer not to antibodies but to affinity reagents, and I think she's right, because it's not just antibodies that can bind specifically some proteins. Actually, you can have all kinds of reagents, usually proteins, often antibodies, but not only, that actually can be used in the same way as antibodies.
So I see another question, I think for Alison mostly, is do you think that in view of these elements, it's possible to act legally at the European level to accelerate the transition?
Sure, no problem. I'll answer that question afterwards. Can I just go back to the poll? I just want to comment on something there as well. Yeah. But in answer to that question, is it possible to act legally? I think at this stage, there's certainly a lot of keen interest in seeing how companies, research institutes, or maybe even member states are responding to the ERLEC ban recommendation. I would say it's early days, but the world is your oyster at this stage. I think, honestly, we need to accept that it's going to take time for member states to get used to it, implement the changes. You know, for example, at the moment, they're still trying to work out how on earth to make this feasible for them. And they're receiving lots of guidance about it. So this is all happening, but you know what it's like at EU level, things happen very slowly. But I do know that there are NGOs that are looking closely at it and starting to ask questions about more recent project license applications that may have appeared in the non-technical summaries, and how they're going to respond to that, I don't know. I think at the moment, questions are being fired and they're waiting for responses, this kind of thing. But I'd imagine if those responses are not adequate, then maybe firmer, more assertive action will be taken. So yeah, like I say, the world is your oyster. I hope that answers your question. I wonder if I may just go back to the poll. Is that okay?
Sure. Yeah.
I was very interested to hear that 2 out of the 14 are producing antibodies in-house. I'm assuming that's animal derived. Obviously, we can't talk to the attendees, but I wanted to let them know if, well, I was interested to know if, in fact, they have access to phage display libraries, and I'd be very interested to explore that further and let them know that it is possible to implement phage display resources and all the technical expertise in-house. And again, if you want to contact me about that, then I can help you with that. Affability is working really hard to help this transition to non-animal derived antibodies, and Affability is producing phage display libraries that they want to make available to people with training, obviously, on how to use them. So get in touch about that if you're interested.
Yeah, I mean, indeed, I cannot speak, but if you feel confident now, you can also write in the chat.
Sure.
And let us know. And I see a question from the panelists. One replied already that they produce antibodies in-house animal-free.
Great.
Yeah, very great. Great. And
it's really encouraging to hear that.
Okay, now I have a long question.
Oh, it's Esther. Hello.
So it seems that to increase the use of non-animal derived antibody and decrease production of non-animal derived polyclonals, there needs to be cooperation with antibody companies, because at the moment it won't work if only one company drops out the polyclonal because other company will sell it. Do you think discussion for the adoption of non-animal derived antibody should also involve more these antibody companies? Who wants to pick it up?
Maybe I can say a few words on that. I think first a rapid answer to Alison. Alison, yes, great. This library is true that having access to phage display libraries is super important for the discovery process. When you're ready to share one, I will be super happy. And we have created one, a phage display library of nanobodies, which we're using currently, which works fine, and we're happy to share it with anybody who's interested. So really,
okay, that's interesting.
Be available.
Is it available without contractual restrictions?
I guess it's coming. It would be by the university, so it would certainly come with something that says if there is a commercial use planned, it should be discussed in due time or something like that. But that would be the super minimal MTA standard. So we would be happy to share it with anybody. I mean, we produced it. There is enough for the whole world to use for a year or 2 or 5. So there's no reason to keep it to ourselves. I think it's important to mention that there is a very big economic aspect into this story of research antibodies. Research antibodies is a big market. There are big companies doing that. There is money flowing in this field. And I'm not complaining. I'm saying we have to realize that we must not be too naive on that, and that we must consider the commercial practical aspects. At any stage, any distribution facility, will have to go private. Any discovery facility, except if it's a super small faculty thing, if it is to grow big enough to be useful by everybody, it will need to get into private company mode. And yes, ideally, there should be collaboration between different companies. We've tried many times to discuss with companies, do you want to even compare your antibody sequences with what we have in the database? And they will not do it. It's too delicate. They're not going to share their sequences. They're afraid they will lose them. They're afraid they will lose control. And it's a difficult question. There is no way for somebody who has an antibody to protect it once the sequence is out.
Yeah, I saw that there was also a question, maybe a bit in the same direction than the one before, from Peter, that is here online. So I don't know, Peter, if you want to ask it directly.
I can do that. I mean, the question is, it's actually very interesting because I was in the European Parliament 2 days ago, and exactly this discussion came up. So we have non-animal derived animal model antibodies now available, and in the field of let's say in vitro tests, if we have an accredited test method, you're basically not allowed to use the respective animal model anymore for this test if you have an accredited test. And couldn't we do something similar in the field of the antibodies? So if we have a non-animal derived antibody available that is validated to a certain extent, can we not, and this goes back to the previous question a little bit, on a legal level, forbid the production and the sale of this respective animal derived antibody?
Well, according to the ECVAM recommendation, then you have the support behind you to certainly argue that case at this point. I think in order for company or a research institution to be using animal-derived antibodies, they have to have really good justification. And it's no longer enough just to write in your project license application in that small little box about the 3 R's, something like it's simply not possible to use alternative methods, they're not ready yet, or etc. etc. Those are the sort of comments we've seen in the past. Now it's necessary and this is what I'm pushing for, and this is my guidance to the member states, it's necessary to provide scientific evidence. I think you need to demonstrate either that you're using, well, firstly, that you have a source, a phage display library source, either your own or in collaboration with another academic group or a commercial institute, something like that. And you need to demonstrate scientifically with data that it wasn't possible to produce that antibody without using animals. If you're using your own library, it needs to be fully characterized, and you need to be able to show that it is fully characterized before you start using it, that you've demonstrated that it can produce a panel of antibodies. This kind of thing is at this stage necessary to justify the use of animals. And I think, as I said in my talk, even if you haven't been able to produce an antibody by using non-animal derived sources, then it's not default necessarily that you will then use an animal derived antibody. You still have to be able to justify it. For example, your issue could be with the antigen, the presentation of the antigen, etc., etc. So yeah, this basically what I'm saying is that you have to have a lot of evidence now. It's quite a formidable undertaking, I think, for a project license applicant. And I think if you are looking at an application, this kind of, as a sort of a lay person or something like that, if you're looking at the non-technical summaries, for example, you can't really see what went into the decision to grant authorization for animal-derived antibody production. However, I think you can ask questions at that stage to the authorizers, why was this particular license granted, and see what they come back at. And if there isn't sufficient evidence, then, I think you may want to go in the direction that you choose to go in.
If I may add one word, Alison is describing the issue of the discovery, which is essential, of course, but think of it also, and maybe that was the meaning of part of your question. Very often there are already available recombinant antibodies against protein X. So it would seem logical to at least test them. To test them first before you go into discovery of something else, injecting a rabbit or whatever it is. And this is our job. This is what we're trying to do, making these available. I mean, the prices are usually between 200 and 400 euros for antibodies that we deliver. This is not going to break the back of any laboratory.
No.
So that should be, yes, I don't understand why it's not mandatory to first try the existing recombinant antibodies before injecting yet another GFP into a rabbit. I mean, there are zillions of anti-GFP antibodies. You don't need a polyclonal.
And I think you have to mention also, and this is in the EURLECVAM recommendation, the fact that over previous 2 years of statistics, I can't remember the figure exactly, but something like 45,000 animals were used for monoclonal antibody production via the ascites method, which is a production method which received attention as when we're looking at replacement far back as 1997, 1998. So it's astounding now that this number of animals is still being used to produce antibodies this way. I keep asking questions and I'm not getting any real answers. I cannot understand why that's still happening when we have tissue culture methods now. Even if you're using hybridomas, I cannot fathom out why that's still happening. It's quite disheartening.
Perhaps for the benefit of the discussion and convincing people, perhaps Pierre, you could recount the numbers of the antibodies already available, more locally and worldwide, so people get an estimate.
So if you go into these big aggregators of commercial antibodies, you find that there are millions of different antibodies available.
I mean, animal-free recombinant.
But if you look, for example, the antibodies that we have that are available is 24,000. So there are 24,000 antibodies that are listed in our database. And I think we have about, we keep thinking there is another, we're missing half of the antibodies. So we keep filling the database, and we estimate now that there might be as many as 50,000 antibodies with published sequences out there that can be made available. Not all of them, as Alison says, were discovered in vitro. Some of them come from hybridoma. But at least when the hybridoma has been there for 20 years, it would be a shame not to sequence it and to use a sequence. I mean, the animal, the involved animal is already long dead. So it would be good to not lose the result of that. But 50,000 antibodies, that covers actually a lot of targets.
So unfortunately, there was a good question in the Q&A, but we need to wrap up. And we can invite you at the end of the meeting. There will be a survey. So maybe you can also ask there some question. We will try to then put you in contact with the speakers. Maybe they can write some lines. So thank you very much. I don't know, Marcel, if maybe you have some final words.
No, I think we really should use all the time on the questions and the talks. And I think this was a particularly rich seminar. And I think one where many can go home to their groups, to their work, to their lab, or to their whatever scientific communities and spread this information. So just remember, it's 50,000 antibodies are available. Pierre is running a facility, it just needs an email, and there are also others, also at the University of Constance, there's one. And so of course, 20,000 would cover the human genome, but there are many, many organisms, of course. So Pierre mentioned Dictyostelium, but if you have anything exotic, perhaps it's even easier to get the recombinant antibody done. So it's an appeal, but I think it's also scientifically extremely interesting because these antibodies are sometimes smaller. So for organoids and other things where it's a matter of penetration and so on, they might even have technical advantages compared to the standard antibodies, and there will be better research results.
Okay. So I'm taking notes of the questions that are open, so we'll try to come back to you. And thank you very much for also to the speakers. It was really interesting, and we had really many questions. And see you next month for the next Talk in 3 Hour webinar.
Yeah, and a hand to our fantastic speakers and to the good questions and discussions.
Thank you for having us.
Thank you.
Don't hesitate to contact us.
Good. Bye-bye. Thank you.
Bye.